Thank you. I'm happy to be here to have this opportunity to talk about a project that we've been working through for a couple of years now. And the focus of the talk today is translating guidelines into effective clinical support and actually evaluating it as well. I have a couple of disclosures to make. One is that I will be discussing a framework that we have created here at CHOP called the Care Assistant, which I do have filed a technology transfer disclosure on. However, there's no patent, no licensing agreement, and I received no money for this invention. I'll also be discussing a vaccine today, and I've been asked to disclose that members of my family have received speaker's fee for HPV vaccine, however, not for the vaccines that are being discussed today. There are two learning objectives today. One is I would like to discuss the process for guideline implementation and measurement. And the second is to explore the strengths and challenges of using electronic health records for this purpose. The focus of my talk will be a case study of our premature infant project, which we've been working on for the past 2 years. We will have a section talking about our approach to guideline implementation, which is informed by some collaborators at Yale involved in the GLIDES project. And we'll also talk about how we viewed clinical decision support more broadly than just alerts. And then finally, I'll close with a fairly large portion of the talk on guideline measurement. A word about the GLIDES project. This is a consortium of healthcare systems that's been funded by the Agency for Healthcare Research and Quality to look in an organized fashion about how you take narrative guideline documents and actually translate that into some kind of a meaningful decision support project. And there have been many collaborators. We work very closely with ECRI Institute in particular, which is actually right up the road in Plymouth Meeting. They're the keepers of the guidelines.gov website at this point and have numerous other activities as well. The aims of the GLIDES project, in addition to what we'll be talking about today, which is using systematic and replicable processes to design, implement, and demonstrate guideline decision support, their secondary aim is also to actually improve the quality of guidelines so that more implementation efforts can succeed. One of the big problems right now is picking a guideline that you can actually implement using any kind of means, much less electronic means. At this point, I'd like to talk about the 3- or 4-step process that we have been going through in a rigorous stepwise fashion to take narrative guidelines and transpose them into clinical executable rules. I realized I didn't have a slide which actually talks about the clinical problem we're addressing, so I'd like to pause here to describe it a little bit. We were focused on premature infants who were born more than 5 weeks before their expected due date. A good number of these infants are actually at high risk for bad outcomes from a particular wintertime virus called RSV. And there is a vaccine that actually helps to prevent hospitalization, intubation, and other bad outcomes that these children might experience. It also happens to be one of the most costly medications that a primary care pediatrician, such as myself, will routinely handle. So there are specific guidelines about the appropriate use of this product to ensure that children who are at highest risk receive the product and that it's not being used unnecessarily due to the cost of the product. So the first step of engaging in a guideline implementation work is to find a good guideline. Interestingly, the guideline that has been published related to this product called palivizumab that we'll be talking about to prevent RSV disease is actually not a guideline, it's a policy statement. However, it actually has some clear recommendations in it that it can be treated as a guideline, but it was actually not published as that. It was actually published as a policy statement because there wasn't really enough evidence base to support using the terminology of a guideline to describe it. We take that narrative documentation. We've been using a tool that's been provided by our collaborators at Yale called the Gem Cutter. This is a tool that allows you to work stepwise through a narrative document and translate it into sort of a pseudocode that can be then used for additional implementation work. In the left-hand pane, there's a highlighted box of text in yellow, which is focused on some eligibility criteria related to infants who may need use of this vaccine. In the middle panel is a tree of information that you have extracted from the document where you go through and you indicate This is a statement that includes some logic criteria as highlighted here. There can be other things that are highlighted, such as this fragment of text is telling you what to do. This fragment of text may be describing a cost-benefit analysis. All of these different components are marked up as you work through the document. And then in the right-hand-most pane, you'll actually see, it's a little hard to see on the screen, but some actual logic which has been highlighted. Here's a little bit more of a clearer output that this gem cutter tool produces, which takes the marked up document and then breaks it out into the input variables that you need to focus on, which is actually a big part of our work, is working on those decision variables, such as here, chronologic age is an input variable, presence or absence of a chronic lung disease condition, and indication of whether or not medical therapy is needed. And there's a lot of text to describe these things. For example, under receives medical therapy, there's descriptions of what kind of medications are actually considered medical therapy for chronic lung disease and should be considered. As we go through this a little bit further, you can then take these decision variables and the way they're connected and actually write executable rules. We've been using actually a Java-based rules engine called Rules, which results in creation of rules that look a little bit like this, which I don't expect people to be able to read on the screen. But there are all of those decision variables from the previous screen, such as presence of chronic lung disease, information about when RSV season starts, how old the child the season starts. Those are all encoded here, and it's almost a line-for-line translation of the pseudocode into actual code that can be executed. I would like to highlight, it's a little too hard to see on the screen, but there's a to-do in here which remains to this day unclarified, because as you write software derived from rules that were written in English language, it calls attention to all the fuzzy boundaries, or sometimes it's described as weasel words, that the guideline authors put in. So in this case, it was actually left entirely unclear in the guideline whether or not when a child reaches the second birthday, if that happens in the middle of the RSV season, do you stop vaccinating them? That was left intentionally blank in the guideline. We chose to err on the side of giving more doses than might have been indicated, and indicate that those children should continue to get doses for the remainder of the RSV season, even if they did reach their birthday. Once you've gone through the guideline and converted everything to executable rules, there's actually a view of the algorithm that the rules engine produces in Java, which each node here represents one decision point related to RSV and the guideline. And we don't really know what this means, but we've been told that if it looks like a flying saucer, then you can be pretty sure you're on the right track. Now we have, at this point in our talk, we've described how to go through taking a guideline and rigorously marking it up and translating it into executable rules. What we haven't yet talked about is what you're going to actually do at the point of care with this guideline. And there was an interesting article published a couple of years ago by Dr. Richardson and some others, really describing the problem that people don't actually even know what is meant by clinical decision support, and that this can become a barrier to the effective implementation of these tools. And they outline three categories of decision support, which I do think are quite useful. One is alerting type of decision support, which is, I think, what most people are familiar with. These are the alerts that pop up in your face and say, do this, don't do that. But there are other types of decision support which we have embraced, particularly for this project. One is workflow decision support, which is a way of easing workflow by improving data entry and documentation tasks or other workflow-related efforts. And then there are also cognitive supports that can provide, for example, some patient management tools, some calculators, or other things that help you with decision making, but actually aren't sort of alerts. They're more tools that you use to help your decision making. At this point, I'd like to acknowledge my colleague, Dean Cherubini, who's in the audience today. He actually did a large amount of work, perhaps the largest amount of work on this project, trying to figure out what the workflow is involved in actually getting doses of the RSV vaccine called palivizumab into the children. And in particular, the alert really only addresses this one aspect of the task, which is identifying based on criteria, who actually is supposed to get the vaccine. And then everything in the middle column and the right-hand column on the screen is actually not described in the guideline, how you're supposed to deal with it. There's a lot of work that goes into dealing with the insurance companies for this expensive product. The sites have to review the charts, they have to submit forms. If they're lucky, they get approved the first time around. If not, there might be an appeal process. And then once the insurance company has granted approval, you need to actually complete a series of 5 doses, and there are many steps, one of which is actually ordering each individual dose from the supplier, getting that into the site, scheduling an appointment for the patient, getting them into the site, and then having somebody with the wherewithal to realize that the dose is there, the child is there, and now is the right time to get the vaccine. So there are a lot of failures that can happen in that right-hand column as the RSV season is in progress. And it was estimated from our qualitative interviews at the sites that a typical requirement per patient is 10 to 20 hours of nurse effort per patient that's eligible just to deal with this workflow over the 6 months. And some of the practices have 50 or more of these patients that they have to manage, so it's a large amount of effort. The low estimates that you're really talking about 1/8 to 1/4 effort for a nurse during the course of the season to actually manage this task. And often sites don't actually have that amount of support available to them. Related to this workflow diagram that we just went through, there are a couple of expanded requirements for decision support that this brought to attention compared to the regular traditional mode of delivering decision support, which is described in the left-hand pane there. We have one patient at a time, we can throw up an alert at the point of care, and you're really just dealing with workflow within an individual encounter. Whereas for RSV, we really needed to help the practices deal with all the patients who were eligible at once. Work needed to begin 1 to 2 months before the patient actually showing up for their first RSV shot. And then the workflow really spans many encounters across a 6-month time period. We also found from some other work that this was not a unique requirement for the RSV vaccine. It actually is almost identical to some tasks that are completed by the ophthalmologists as they're tracking the patients who are at higher risk for eye disease from prematurity, and is also very similar to the tasks that are required by allergists who are performing immunotherapy on their patients. So we have here, this is just a slide about the framework that we've used to deal with these additional requirements, which are not really addressed by the tools that are provided by electronic health record vendors. So we expanded the framework out of necessity to actually provide these additional features. And in particular, the 5 items that we added with the framework is an ability to actually mine the electronic health record data in real time, to incorporate the use of a formal rules engine as opposed to the stripped down logic that the vendor typically allows you to do. We needed to automate some EHR functionality so that if there was something that the user needed to go click, 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 click 10 times to get through something, that we could sort of do those clicks for them in the workflow. We also needed to pull in some external materials into the electronic health records, such as literature and other documents. And we also had to put together new ways of displaying information to present all this in a succinct format to the user. And listed on the right are a few projects that we've already completed, one of which is the premature infant project. And then we have a couple of projects ongoing where we're actually trying to reach out directly to patients with these tools and engage them in shared decision making. And then we're also involved in a genome sequencing project, which is getting very excited to figure out how we're going to incorporate that information into the point of care. This is a screenshot of what the nurses who are coordinating the RSV workflow actually see when they're trying to deal with sort of all the patients at once in the practice. This is just a handful of patients. This uses an existing feature in our vendor called the patient list feature, but we had to apply a lot of Band-Aids and paper clips to kind of enhance it to do the additional tasks that were needed, such as reporting eligibility criteria and tracking the status of the patient through the season, and then things such as knowing what insurance company we're dealing with is very important for this, because each insurance company has their own forms. The nurses have some additional tools that we provided as part of this intervention. which include, at the top left, there's the AAP policy statement information, and we actually produce for them a printed document that allows them to get the key variables for the insurance paperwork. We don't actually generate the insurance forms, but we do try to make it easy for the sites to generate it. And then at the bottom, there's a dose scheduler, and it does a few things, one of which is to predict the weight of the patient at the time the dose is due, because they need to know how much of the vaccine to order for the upcoming dose, because it's a weight-based dose calculation, unlike other vaccines. And then there's information about when is the ideal window of time to give the dose, and whether or not an appointment has already been scheduled or not. So that's the nursing view of the decision support tool. And here's what we did for the physicians. And to use Dean's words, we tried to reduce the guidance for physicians down to caveman speak, to try to keep it as succinct as possible, because we're trying to present a lot of information, and doctors don't like to read lengthy descriptions. They just want to know what the point of the matter is. So highlighted here is the way we describe growth, which is not related to the Synergist project per se, but is related work. We just summarize that as low weight. And then we have linked to some additional resources, such as here we're showing the detailed assessment of growth. And at the bottom half of the screen are some tools to do nutrition calculation for oral nutrition for these kids. So if there is a problem related to the growth, you can actually do some quick calculations to figure out approximately how many calories the child is getting. And there's actually no rules here telling people what to do. This is just a calculator so that people can figure out what is currently happening, and then look at how many calories a child would be getting with some alternate regimens. If we had structured guidelines that we could have digested to produce specific recommendations, we could have incorporated that here. But we felt it was useful just to give them the cognitive support of being able to calculate nutrition and figuring out what would be an appropriate target for the patient. We also provide a little bit of information for the families as part of this project. Again, a lot of this isn't specific to the RSV project, but it's the total package of information that is needed to take care of premature infants. There's a personalized report that each family gets at each visit, which describes their growth status, development status, a few highlights about nutrition, such as highlighted here is the point that they should not start baby food until a particular time point here, saying approximately 5 weeks. And a few other things that are personalized specifically to the individual patient to give specific and concrete guidance directly to the families. And this is provided as a printed document. We also provided printed documents related to content on our own intranet, such as shown here is how to prepare infant formula. This type of information is what the providers often most appreciate about these projects, is being able to actually get at the information that's already available online in a more streamlined fashion. At this point, I'd like to switch to talk a little bit about how we decide whether or not we have succeeded with our intervention. The guidelines are often very focused on telling us what should be done for a particular clinical situation, but they don't actually tell you how to decide whether you succeeded or not. Often you can sort of come up with a reasonable assessment based on what the recommendation is saying, but it actually may be a little difficult to choose a metric that's actually both meaningful and that can be measured, so that at the end of the intervention you can decide whether or not your implementation would actually succeed. So we're going to explore these issues, and actually we had an hour-and-a-half meeting just this morning talking about the evaluation of the RSV season. And we've had about, I would say, 20 such hour-and-a-half discussions. So this is a lot of work just to decide how to evaluate it. And we're trying to figure out all of this before we actually really look at the data, because we want to be able to be clear that says we pre-specified this is how we're going to define the success of the intervention. Today I'm not going to discuss choice of design, either experimental or quasi-experimental, because that pushes the boundaries of my knowledge a little too much. But I can try to address that in question and answer if desired. When you're doing an intervention in electronic health records, such as we are doing, one of the problems is you're going to decide the success of that intervention based on data in the same system. And there are a few pitfalls that need to be kept in mind as you're looking at the electronic health record data, one of which is the accuracy of the data. There can be two common problems with accuracy. One is errors related to the human assessment, such as the classic example is murmur. Presence or absence of a murmur is most likely predicted by involvement of a cardiologist, not presence or absence of murmur in the child's chest, as well as data limitations such as typical electronic health records may not allow you to capture even basic facts such as the severity of a patient's asthma. Another concern which I think confuses many researchers is the fact that the data change every day. And whether you extract your data set today versus a week from now will result in different results, even though your study period may be something in the past. And a couple of reasons is that medical record numbers will change over time, largely due to patients showing up at a new care location and being registered under a duplicate medical record number because the registrar didn't find them. Maybe they have a hyphenated last name, and they looked up the hyphenated last name in the reverse order. Any number of things can happen that result in a patient being assigned a new medical record number when they show up at a new care location. And then eventually, hopefully, somebody realizes that the same child is being described under two medical record numbers, and then those two charts are merged. And what happens is it appears that somebody disappeared from your cohort because likely you had that child in your cohort twice, even though they were only one person. So what I usually tell people is if we've re-extracted the data, please tell me if some of your retrospective data changed by more than about 1%, or if it changed by exactly 0%. That would be a little surprising because almost always there's a little bit of change once you re-extract the data. And if there was absolutely no change at all, that suggests that maybe we didn't actually extract the data again. We may have accidentally resent the original database. The other thing that happens is clinicians rewrite history every day. Sometimes this is, well, always it's well intended, but sometimes this is a result of new information arriving in the practice, such as a new immunization history, which shows that, for example, maybe the child already got the palivizumab Synagis shot, either at the hospital or at some other location, and we didn't know about it at first, but now we do. So we realize after the fact that the practice made the right decision to not give the vaccine because it had already been given. Or it can be a matter of error correction, where somebody realizes a couple days later that, oh, they had told the patient to start this medication and they phoned it into the pharmacy because Epic was down, and now they realize, oh, I have to go back and abstract that in. So all those things can actually change the data in very unexpected ways over time. So the consistency of one data extraction versus the next can differ a lot. And then sometimes there may be information that you simply can't get at because it still lives in a paper record and is not actually available by any automated extraction means. For example, ADHD assessments in our practice are most often collected in a paper format. These are questionnaires to assess how a child is doing at school with paying attention and some other characteristics. And it's usually delivered to the school in a printed document. The teacher circles some numbers, faxes it back to the office. Physician may or may not describe in their note what they found on that report, and then the document is either put in a paper record or scanned in, but it's basically not available in any way, shape, or form for electronic access. And also we get letters often from outside consultants, often they're handwritten letters, and those sorts of healthcare details are just not available. So for example, for our ophthalmology-related project with eye disease, we cannot really make one of our outcomes to be whether or not the child actually saw an eye doctor because it's too hard for us to determine in any reliable fashion whether or not that actually happened. So you have to look at other things that you can measure. Okay, for the remainder here, I'd like to focus back on how we approach these issues for the RSV vaccine, which is palivizumab. Using the approaches we were talking about earlier, we decided to take a comprehensive quality improvement project, which fortunately, actually, the CARE Network was already interested in doing. One thing that cannot be stated often enough is to choose a project that the clinicians actually want to participate in. And we were very fortunate because this was a big issue amongst the CARE Network. There had been sort of a set of work group meetings amongst some clinicians talking about how to approach palivizumab administration as a QI project. And we started this initiative in spring of 2011 with a goal of improving the administration in the upcoming season, which is now in the past. But even before we actually started really to do the intervention, we took a look at the baseline data to figure out where the important intervention opportunities were at. And as there was a brainstorming activity to figure out where we thought the problems were, there were many questions. We realized even the simple questions we didn't know the answers to. For example, we didn't know who should have actually gotten palivizumab in the past season. We didn't even know who did get it. We don't know if they got their doses on time. We don't know if the family was refusing doses. And another concern that was raised, which hasn't yet become too much of an issue, is that there's a movement to do these vaccines actually at home. And information about those doses may not be available to the healthcare system because a home nurse goes out, usually sent out by the insurance company, gives the vaccine at home, and if we're lucky, we get a phone call from the insurance company saying that it happened so that we can put it in the chart. And many actually view this as an ideal situation because the last thing you want to do in the flu and RSV season is bring an at-risk child into your office and surround them by a ton of kids who already have RSV and say, we're going to protect you from RSV now with this vaccine. Gosh, I hope it works. Because there's plenty of opportunity to get exposed to the disease while you're in the office. So as I'd like to now go through, this is a bit of a tongue-in-cheek exploration of how we looked at the data. I'm actually the report writers, so I actually did know how to ask the questions correctly, but I'd like to highlight how a typical interaction goes when one is working with reporting people who may not be able to read your mind and understand what exactly your question is. So I began my discussion with dear report writer. We'll call him Mark, because he's usually the person I turn to for these quick questions. Mark, quick question. Who got palivizumab last year? And I get back a quick answer, 872 patients. And I said, gee, that seems a little higher than I was expecting. And my report writer gives me a little more details and says, yes, 872 distinct patients received a dose between January 2010 and December 2010. Oh, by the way, should we exclude those doses that were deleted? And I say, oh, yes, absolutely, zoinks. My son likes Scooby-Doo, so get a few Scooby-Doo references here. Yes, please exclude those deleted doses. And what I really meant was not last calendar year, but I want to know about the last RSV season, which was November 2010 through March 31st, 2011. So I get a new answer, 639 patients. Thanks, that sounds more believable. How many doses were given in total amongst those patients? Those patients received 1,782 doses. Nice, simple answer. But when you do the arithmetic, you realize, gee, that's only 2.8 doses per patient. And what we were talking about earlier in the guideline is that typically patients are eligible for 5 doses if they're eligible for this. So why so few? Then we might ask the question, wait a minute, when are these doses actually being given? And the report writer at this point starts to get a bit cranky and says, hey, you said this was a quick question. But here you go, it was given a lot of different places. There were this many doses given in primary care network, 1 dose in dermatology, 1 dose in occupational therapy, and 452 doses were given elsewhere. Huh? Dermatology? Why would they be giving Synagis? That doesn't make any sense. They don't give that vaccine. The reporter writer said, oh, yes, indeed, the patient named Test Buddy got 1 dose from dermatology. I can't make this stuff up. This really happens. And I'm sure I was kind of writing these reports as I was going through this experiment myself and what an interaction would be. And I did find a dose given in dermatology. And this happens just all the time. We forget about the test patients. And sometimes I'll say, if you don't have a test patient show up on your report by accident, whatever question you're asking, it must not be very important. Because our test patients receive the most and the worst health care in our organization. So you always have to keep those in mind. So we have a little profanity, which we keep inside our own head because it's a recorded session. And we say, please exclude the test patients. I'm really only interested in those doses that were given in the care network locations. Thank you. So the new answer is that actually only 430 patients were getting vaccine in the care network, and they received over 1,300 doses. So this is the typical set of basic criteria that needs to be included in any data-related question. And of course, it often gets much more complicated. Often people know what they're asking for, and I would say about 80% of the questions that we receive on our data reporting group, they'll usually specify up front what the when is, what the time period is. But it's actually surprisingly often how they haven't thought about how complicated our healthcare system is and what actually portions of our healthcare system are they actually interested in measuring. And then also it's really our job on the reporting side to remember to keep the test patients out of the data set unless for some reason we're specifically wanting to look at a test patient. Now we'll move on from looking at who actually got doses and go to the next level of complexity to say is how many of these kids actually got their doses on time. And the interesting thing about the guideline from the AAP is that it just says doses should be given monthly. It doesn't give any guidance about how frequently to give. So monthly to me means November, December, January, February, March, which is actually works not too badly. But we actually had to go to the manufacturer of the product and ask, what is actually the dosing? The package insert didn't really have it either, as I recall. And what they told us is this is how the product was evaluated in the randomized clinical trial. And they gave us the boundaries. It was actually a different interval for the first dose than the second to the fifth doses. It's roughly a 25 to 35 day window is how it worked out. But none of that was stated in anywhere that's actually readily accessible. Presumably most practices are doing the right thing and are correctly interpreting what monthly means. It doesn't mean you give it on November 30th and then on December 1st. It means something like a month should be between them. So we looked at, within each care location, how often did a subsequent dose get given within 31 days of the prior dose. And this is what we got in a graphical form. Shown in green is the happy result where the proportion of doses were given, the proportion of doses that were given within 31 days was high. We sort of gave a grace period of an additional 6 days, which is shown in orange. And then people who really were outside of that window, which would have been, I guess, like 40 days or more between doses, were shown in red. And we can see that the glass is slightly more than half full, that overall, of those who were getting multiple doses, probably over half of them at most of the sites were getting them within an ideal time period. However, there's also still a lot of room for improvement. We then went to the really hard part, and this took a lot of work, was to actually figure out who should have gotten the doses. So it's nice to talk about the people who actually did get the doses, but we were also very worried about those who should have but did not get any doses. And this was actually quite challenging. So we decided to look at a specific subgroup that we knew we could define fairly well. And that is a subgroup of children who had established health care before the start of the RSV season, who we could easily prove were eligible for at least 5 doses based on their prematurity alone. So we had to take the AAP policy that we translated for our guidelines and retranslate it to be a query for the report writers. And this is how we described it. So we looked at gestational age criteria within a certain birth date range and tried to keep it as simple as we could. And then we reported of that how many of the children who should have gotten 5 doses actually did get 5 doses. And now we have a glass which is arguably much less than half full. So all the, each practice is shown separately here. The numbers at the bottom represent how many patients were eligible at each practice to receive the dose. And then shown in green is the proportion at each site who got 5 doses. We gave partial credit for 4 doses because 4 out of 5 isn't too bad. And then kids who got 1 to 3 doses are shown in red. And then the empty spaces represent kids who should have gotten 5 doses, but in fact got 0 doses. This, looking at that lovely slide, which the statistician was ecstatic about, he said, this is great. You're starting with a really low baseline. It's unlikely that you'll accidentally make things worse, and your intervention will probably make things better. And so that's great. I wanted to use this as a moment to talk about another pitfall with data, which is how do we know that we actually detected the right group of kids who are at risk? Because you have sort of this self-fulfilling prophecy that happens where your better providers, amongst the better things that they do, is they do a better job documenting in the chart, which makes it possible for us to better identify their patients as being eligible, and then presumably they are also doing a better job of identifying addressing these children's health needs. So you can lull yourself into a false sense of security, although I don't think one would have felt too secure from the previous slide. But it can happen that you can get this apparently good result and conclude incorrectly that there is no problem because you failed to measure the problem because it's essentially undetectable with the existing data. And also I just want to take a moment to ask, where will you take your premature baby? So this is the Child Care Network. I've shown this slide to the medical director of the Care Network and asked permission several times. And, do you really mind me showing this slide? Because it's exciting for us to have opportunities for improvement, but also it can be a little unsettling when you may feel that you expect the healthcare system to be doing better. And one thing I just want to point out is that one should not pass judgment on Group A. They only had a couple of kids. They are really no different from the sites in Group D or the single site in the middle that got it half right. It's just that the practices in Group D, I would presume, got lucky, and Group A got unlucky. But there is something different going on between Groups B and C. And so we tried to drill down a little bit and take a look at that. So of the 46 premature infants in that group who we could easily prove with a simple query were eligible to receive 5 doses and had every opportunity to get them, only 35% of them actually got even 4 doses, which was great news for the QI team because then we can have a nice positive result, hopefully, at the end of the study. But we did consider some possibilities of how this might actually be incorrect. One is that we might have been detecting a cohort that wasn't really eligible. One is we might not have been detecting the outcome correctly. For example, if a lot of patients were getting their doses outside the healthcare system, we wouldn't know it. And the other possibility is that it is the truth, but the problems are not ones that can be remedied by the healthcare system, such as patients not showing up for appointments. To look at the question of whether or not we were actually able to identify the cohort at risk, that can be very hard to do. One thing I attempted to do here is to look at, by site, of the kids who actually got the doses, how many had a gestational age on file, to see if those who were getting the doses were actually doses that we would have identified. And this still has a little bit of a, if you look for the kids who are getting good health care, you'll find the kids who have good documentation. But we were reassured to find that essentially everybody in this cohort did have a gestational age documented. This is an optional field that's filled out often at the time of the initial visit, and one always has to worry about whether or not those fields are actually available. Just the little red tips on the top of some of the bars are that subgroup of patients who did not actually have a gestational age on file. So we're actually fairly encouraged about the quality of the gestational age, although one could still have some worries about that. The other thing you can do by looking at your baseline data before you start the intervention, especially if you're doing a randomized clinical trial and you can compare cases and controls, is you can actually ask the question about ways to improve the quality of the documentation so you can better measure your outcome. And in fact, there were some efforts, nothing heavy-handed, but there was a lot of communication about when you're seeing these children who are chronically ill, please put some specific information on the problem list, like indicate if they have chronic lung disease or if they have a clinically important cardiac problem or other problems such as airway issues or neuromuscular problems or immune problems. And it was not uncommon for kids to have pretty significant healthcare problems and have it not really very well described in the problem list. So that was to the extent that we encouraged, or the Care Network was encouraged to improve documentation, was really to put some descriptive, specific details on the problem list about these at-risk kids. One can also look at other documentation that might help with identifying these kids or improving documentation or even assessing the quality of the documentation. For example, we often look at medications and say, okay, we've got 1,000 kids who are receiving flow vent inhalers, and of these 1,000 kids, only 200 of them have an asthma diagnosis. at these 5 care locations. So that would indicate that, for example, the problem list might not be accurately reflecting the disease state of the patients, because they're clearly getting a medication for a specific problem, but the chart is otherwise silent about the presence of that problem. So you can use these strategies to try to iteratively improve documentation by looking for specific recurring problems. And also, we read, I think it would be an understatement to say, a lot of charts as we went through this process of trying to refine our methods and queries to try to make sure we were actually detecting the truth. And that's something we encourage all investigators to do, is if you ever get a number that seems either too good to be true or too bad to be true, look at a lot of charts before you start writing papers based on those numbers. And even if it looks about like what you expected, it's still worth looking at a handful of charts just to make sure you don't have big systematic errors in the way you classify your patients. For the baseline season, we did go through the charts and we asked further a question which simply could not be answered by electronic means. We asked the question, why did the children actually miss the doses? And we found that the #1 reason was, in fact, that the clinicians had not actually recognized that the child was eligible. Or if there was recognition, there was nothing in the chart documenting that the children should have received the palivizumab vaccine. The second most common was that there was clearly documentation in the chart that the child should be getting the vaccine. However, there was actually a no-show problem, especially in some of the urban practices where there are more of an underserved community, where the healthcare system was making a pretty good effort to do everything correctly, but the patient just didn't show up. And you can't hop in the car, throw an RN in the car, drive out to their home address and give them the vaccine. The way our healthcare system works nowadays is you need them to show up and darken our door. There was some problem that was arguably could be improved by the healthcare system, which is if a child no-showed, often, at least in 13 cases, there was no indication of any attempt to reschedule that missed visit. And then sometimes, and quite distressingly, we could clearly see documentation that indicated that the child was in the office, the vaccine was in the office, they were eligible for the vaccine, there were no contraindications at that visit, yet the vaccine was not given in 10 cases. And only in a couple cases, actually, do we find that insurance or denial or family refusal was really the problem in this cohort. Now, this is keeping in mind that the cohort we looked at were kids where we could easily prove they were eligible. So the insurance company was pretty likely to agree as well that they were eligible. So one important limitation that we knew affected our data quality is that at every single site, and thank you, Dean, for taking some photographs of this artifact, every single site has these Synagis binders. And a few examples are shown here of what was found across the various care locations of these shadow binders that existed. And on the left hand, and I love it, they've got 4 binders to manage their patients. A lot of paper that is not accessible in any way to us unless you go to the site and say, show me your Synagis binder. And a little bit before this, but it was a fairly depressing but not untrue article that was written, which is excerpted here with a little quotation, that essentially the formal chart is just a task, that the formal chart being the electronic chart, is just a task that needs to be completed. This clinician felt it provides little direct benefit, and in fact what has happened is these shadow charts, such as the Synagis binders, have actually become, in some cases, the real chart, and is the artifact of supporting the clinical work. And that was definitely true for the Synagis vaccine situation, but I don't think is actually true entirely of electronic health records, but that definitely rang true for the problem we were having with Synagis. Nevertheless, with all these limitations and the couple minutes remaining, I wanted to just share a couple of the results that we found after we undertook this work. In the post-intervention period, this data was some very preliminary data. We looked at how the season got started. We have some publication pending results, which I haven't reported here, which tells a similar story. But we had a pretty good improvement in the ability of the practices to get a timely start to the Synagis season. So amongst the cohort at risk in the pre-intervention season, only about 3/4 had even received a single dose by December 31st, and we were up at about 83% in the post-intervention period. If you looked at, actually we did have some year-end statistics here. By the end of the year, 65% of the kids who should have gotten 5 doses got at least 4 in the post-intervention period, compared to 54% previously, representing a little over 10% difference in the proportions. And we're now very rigorously looking at whether or not we did change some of the failure modes that were experienced. And it seems pretty clear that our intervention, if nothing else, had at least almost eliminated the problem where the clinicians didn't recognize that the child was eligible. We were very good at getting rid of that problem. Unfortunately, some other problems became more prevalent, such as specifically the scheduling of patients will almost certainly emerge as an area where ongoing work is required. Another thing that I think we were pretty excited about is, at least at some care locations, those big, huge binders are becoming less important in the synergist workflow. Shown here in the screenshot, again, thank you, Dean, for taking pictures, is what remains of the shadow chart for one care location, which is a thin little manila binder, and shown on the screen in the background there is actually the Synergist RSV vaccine tool that was being used by the practice. So we're hopeful that we've actually tried to at least move the information into the chart, as well as actually helping to reduce an important healthcare problem. In summary, there are a few perks and pitfalls that I'd like to re-emphasize. One is that, in fact, the EHR can be used to implement guideline-based care in creative and effective ways. Sometimes it requires some special tools beyond what the vendor provides, but it is possible. Measurement can be done, but it is challenging, and it's important to look for corroborating evidence if the data are suspicious or missing, and it can be important to look at free text as well. And as I say at the end of every presentation I give away to data, is you really have to at least look at some charts to make sure that you are actually getting the accurate information in your data. And I would add, as a footnote for this project, you should also look for the shadow charts too, if you know that they are there. I'd like to acknowledge many people, including our funding, which was a combination of funding from the National Library of Medicine as well as AHRQ, and then a team of many individuals, some of whom are in the audience, to which I'm very thankful for all the help with this very complicated and exciting project. And at this point, I'd like to thank you very much for your attention and invite any questions.